In this video, we will be talking about the large biological molecules, or in other words, the macromolecules. I do have to change one thing. This is supposed to be nucleic. Okay, so basically, we have three macromolecules. They are true polymers, and for lipids, they are not true polymers. Although we have four of them, they are all classified as large biological molecules. We only have three macromolecules. So these are the ones: carbohydrates, proteins, and nucleic acids. So that's it. The only difference between these three and lipids is that um, for these are macromolecules because they are big enough and they are considered true polymers. For lipids, they are not considered true polymers. So cells can make many different polymers by linking a limited set of monomers into diverse sequences. So this is basically, I hope to say that it's a flowchart, but it doesn't look like much. But yeah, for carbohydrates, there are three types: monosaccharides, disaccharides, and polysaccharides. In some textbooks, it might be apparent that there is another term called oligosaccharides. So let me explain. For monosaccharides, we have one, coming from the word mono, from the prefix mono. There's only one one simple sugar. And for disaccharide, coming from the prefix di, it means double sugar, so it will always be two. For oligosaccharides, we have 3 to 9, and for poly, 10 to more. But particularly as per the Campbell Biology textbook, oligosaccharides are already part of poly. So for mono, we have glucose. For disaccharide, we have sucrose, which is basically glucose plus fructose. While for polysaccharides, again, it's 3 to more. So we have roughly 100 to 1,000 monosaccharides joined together by what is called a glycosidic linkage, or in other words, glycosidic bonding. That's the bonding of carbohydrate molecules, particularly monosaccharides. So let's go first with monosaccharides. So for glucose in particular, they are major nutrients required to fuel cellular work. For instance, cellular respiration. And for the carbon skeletons, they are equal to raw materials. They are needed for the creation of types of smaller organic molecules. So yes, glucose can become monomers for disaccharides or polysaccharides. Again, monosaccharides, if you add these molecules together, you will have disaccharides. If you add one together, that makes two, and more than two makes polysaccharides. So criteria for classification of monosaccharides is basically the location of the carbonyl group, aldose or ketose. For aldose, you will see it at the very edge of the C skeleton, or the carbon skeleton. And for the ketose, you will see it within the carbon skeleton. As you can see here, the word dulo means at the very edge. That's very edge in Filipino. And for number two, the second criteria is the size of the carbon skeleton. 5 to 6 carbon sugars form rings since it is structurally stable. And for number three, spatial arrangement around asymmetric carbons. For trademarks, we have two things: carbonyl group and hydroxyl groups. Now, for disaccharides, we have, this is again called in other words, double sugar. The most common example is sucrose, and this is basically two monosaccharides joined by glycosidic linkage. As this is exactly the same process with polysaccharides, glycosidic bonding, and this results to disaccharide synthesis. So we have three examples of disaccharides. We have lactose, sucrose, and maltose. Lactose is synthesized by the addition or by the sum of galactose and the glucose molecule. For sucrose, it's fructose plus glucose, as I mentioned earlier. And for maltose, this is particularly an ingredient in making beer. It's basically two glucose molecules added together. Now, let's go with polysaccharides. So there are two things that fall under polysaccharides. We have storage polysaccharides and structure polysaccharides. For storage, um, literally from the word storage, basically serves as storage material hydrolyzed to cater monosaccharides to cells. Again, cells need to, in order for them to function, they need monosaccharides, they need fuel. Our body needs carbohydrates so that we basically carbohydrates are an important source of instant energy. So yeah, athletes need more carbohydrates than the normal person. Picture that. And an example of a storage polysaccharide is starch. This is the polymer of glucose monomers. Uh, this is basically the one associated with the storage of um, food for plants. Plants store these as plastids. They withdraw sugar by hydrolysis. More on this later, the term hydrolysis, as well as another term that's related to it, actually the opposite of hydrolysis, which is called the dehydration reaction. I think it's the next page, yeah. Synthesis allows plants to stockpile surplus glucose. So it's basically stored energy. One thing about carbohydrates is that, particularly for humans, if there is an excess amount of sugar, that eventually becomes a lipid, becomes a fat. So, yeah, you don't really have to know the specific parts, but yeah, review at your own pace. Review according to what you have to prepare for. So for starch, we have 1 to 4 linkages, glycosidic linkages. The simplest form is called amylose, and the complex form is called amylopectin. The reason why amylose is healthier than amylopectin, it's something associated with the idea that when you consume amylose, you have higher resistance to starch, if I'm not mistaken. So yeah. Another thing is that glycogen, this is for animals. If starch is for the food storage of plants, glycogen is the food storage for animals. Criteria for classification, the monosaccharides present in the polysaccharide structure and the position of its glycosidic linkages. Now, let's go with the structure of polysaccharides, the second type. Literally from the word structural, these polysaccharides serve as a building material for structures that protect the cell or the whole organism. Organisms build strong materials from these. So we know that starch is the food storage for plant cells, and of course we also know that plant cells have cell walls. Now you might wonder what these cell walls are made of. So for plants, its cell walls are made out of cellulose. So 1 to 4 linkages, and they are insoluble fiber. Cellulose in particular, human digestive enzymes cannot digest cellulose. That's the reason why sometimes there this usually happens at least at some point in your life. Uh, when you defecate, your stool looks like what you ate. The reason for that is um, your digestive enzymes are not able to consume the food properly. So what you release out of your system is still the same as how it looked like when you ate it. So for chitin is a carbohydrate used by arthropods to build their exoskeletons. And uh, chitin is also a material used for for the cell walls of fungi. Alright, more on dehydration reaction and hydrolysis. Um, these are simplified terms that we have to associate it with, that you have that you have to associate with. So ketose is basically ketone sugar. I think it's made out of fructose. Aldose is basically aldehyde sugar. Um, it's made out of glucose. Well, basically, for dehydration reaction, this is the formation of glucose monomers, and at the same time, uh, a water molecule is removed, if I'm not mistaken. Let me double check. There, the loss of a water molecule. And the opposite of a dehydration reaction is um, basically hydrolysis. This is the reverse of condensation reactions. Condensation, uh, more specifically, dehydration reaction that I just mentioned. So, yeah, adding water to one large molecule. There, addition of water. So, one real life application of hydrolysis is in your stomach. Your stomach is capable of hydrolyzing the food that you ate using its hydrochloric acid. It's the process of digestion is associated with a concept that you'll be knowing in the chapter of proteins, particularly proteolysis. But more on that soon. Most animals have enzymes that can hydrolyze plant starch, making glucose available for cell nutrition. So, starch sources in human diet include grains and potato tubers. Grains, wheat, maize, rice, and grasses, many more. Uh, so, vertebrates store glycogen in liver cells and muscle cells. So, there's a difference. Um, this is supposed to be associated with these. Glycogen is only for animal cells, as you can see here. But for, how do I say this? Amylose and amylopectin. So basically forms that you see in sugary foods such as pretzels, bread, etc. I remember answering this particular question in my test just a few minutes ago, and yeah, I didn't get the right answer. So I know that there are certain ideas here that you think are not supposed to be associated with each other, but particularly for my quiz, they're supposed to be associated. So just learn at your own pace and according to what you have to review. Stored fuel cannot sustain some animals for long. So that's the reason why low carbohydrate diets can cause a higher tendency for weakness and fatigue. So for polysaccharides, we're going to compare starch and cellulose. Um, they are both utilized by the plant cell. Starch is for the storage of sugar, for fuel, storage of fuel, and cellulose is for it's basically the material for the plant's cell wall. So when glucose forms a ring, hydroxyl group is either above or below. So yeah, you might want to associate starch with helical. Cellulose is straight. So glucose monomers of starch are all in alpha configuration. On the contrary, glucose monomers of cellulose are all in beta configuration. Starch and cellulose, they have different glycosidic linkages, which means they also have distinct 3D shapes. So enzymes that digest alpha linkages of starch by hydrolysis cannot hydrolyze beta linkages of cellulose. So what this sentence basically means is that our enzymes as humans, we can only digest particular food, but whatever that has content of dietary fiber, since we don't contain what is called cellulase, cellulase is an enzyme that digests cellulose. We're not able to digest cellulose. So cellulose is basically dietary fiber. So for examples include cows are able to digest cellulose in hay or grass since they have enzymes inside them that are able to digest cellulose. Basically, those enzymes are called cellulase. They can be prokaryotes, protists, etc. And for termites, they can also digest the same thing in wood. You know, humans cannot eat wood. That's common sense. But for termites, they're able to digest wood. The cellulose content in wood is digested by its prokaryotes and protists. And for fungi, cellulose is in the soil, so fungi feeds on soil, I guess. And yeah, cellulose versus chitin. The only difference is that, they're both materials for the making of cell wall, but cellulose is more on plant, more on plant cell. Chitin is more on fungi. And there's another thing. There's something called peptidoglycan. This one is the material that you use for making the cell wall of what is called Bedelivibrio bacteriovorus or prokaryotes. So yeah. Anyways, this is the next following pages would be a summary. I guess not a summary, but a rephrase of what is mentioned in the Edpuzzle video called Carbohydrates and Sugars. This video is from Osmosis, by the way. Our teacher only posted it on Edpuzzle for us to study. So for carbohydrates, they are simple sugars, or they are complex carbohydrates. They provide calories and energy. Uh, I would be redundant if I continue discussing everything that's mentioned here, but yeah, it's already mentioned in the osmosis video, but yeah. Carbohydrates divided into two things, natural sugars and added sugars. So yeah, for added sugars, we all know that these foods are usually processed, literally added sugar. So the sugar content is unnecessary, but it's added anyways. So sugar refers to a family of saccharides. Again, what I mentioned earlier, monosaccharide is one sugar molecule, di is two sugar molecule, oligo is 3 to 9, polysaccharide is 10 to more. And for glucose, it's a monosaccharide. It is the most important member of the sugar family and is one of the main sources of calories. It can cross the blood-brain barrier and in turn nourish the brain. For fructose, it's most commonly found in honey, fruits, and root vegetables. And for galactose, it's basically milk sugar. Oh yeah, these are all monosaccharides. For disaccharides, addition of one, the sum of two glucose, two molecules, two monosaccharides, that makes a disaccharide. Okay. Complex carbohydrates, we have oligo and poly. For polysaccharides, we have starches and cellulose. Starches, cellulose, dietary fiber. I mentioned to you earlier that cellulose, an example of that is dietary fiber. Human intestinal enzymes cannot break down. Therefore, they can pass through the small intestine undigested. They are broken down a bit by bacteria in the large intestine. Large intestine collects excess water and contributes to the formation of feces. And eventually, it ends up as bulk matter in stool. So, one good thing about dietary fiber is that it increases stool weight, which in turn reduces the chances for you to be constipated. And an example for dietary fiber is beta-glucan for a reduction in the tendency for you to get a heart disease. Anything that's related to cardiovascular. And for starches, human intestinal enzymes can break down. It's one important source of calories, and it's not sweet. So I guess you can call starch as a natural sugar. I'm not sure, but yeah. Come to ponder about it. And yeah, dietary fiber is critical since they also slow absorption of simple sugars in the small intestine. So it also in turn maintains blood glucose. So there are three advantages given by dietary fiber. We are blessed to not have the digestive enzyme to transform dietary fiber normally. So yeah, monosaccharides link together through what is called a glycosidic bonding. Earlier, it was mentioned as glycosidic linkages. I guess they're the same. I'm pretty sure, but yeah. Glycosidic bonding is basically the bonding of OH group with hydrogen between two monosaccharides, and it forms a water molecule which goes away. This water molecule goes away in the event of a dehydration reaction. So, yeah. Different amylases break down different linkages. So, amylase, this is an enzyme that we have as humans. They break down polysaccharides such as starch into smaller units. We can break down starch, but we cannot break down cellulose. Secondly, lactase breaks down lactose. Sucrase breaks down sucrose. Maltase breaks down maltose. Um, as you may have observed, any digestive enzyme has the suffix ase. So yeah, you might want to take note of that. These monosaccharides, glucose, fructose, and galactose, they enter the bloodstream to get used by the body in many ways. So when glucose levels increase after eating, the pancreas releases hormone insulin, which moves the glucose into all cells for energy. So glycogenesis, the creation of glucose molecules, stimulates the liver to store glucose as glycogen. It promotes fat and protein synthesis. And glucose molecules get linked together by alpha 1 to 4 and alpha 1 to 6 glycosidic bonds as glycogen, a form of polysaccharide. So, if I'm not mistaken, insulin is um, something that's created by the liver, if I'm not mistaken. Let me double check. What secretes insulin? I think it's pancreas. Ah, yeah, pancreas. Pancreatic cells secrete insulin. It regulates your blood sugar, basically. Oh, yeah, should have understood this part. Anyways, in the liver, metabolism of galactose by the liver enzyme converts it to glucose. And fructose, through the liver enzyme, it becomes 2 to 3 carbon molecules, which undergoes glycolysis to generate energy. When energy is needed, all three monosaccharides are metabolized through glycolysis, then the citric acid cycle, and then oxidative phosphorylation. Sorry about that. So ultimately, all digestive carbohydrates, either simple sugars or starches, they are broken down into monosaccharides for the purpose of immediate energy use or storage. So this is one thing that you have to know about carbohydrates. Carbohydrates, immediate energy use. The sugar is used for instant energy. So this is the one. And for storage, excess sugar tends to be lipid, turns to convert as a lipid. So yeah, this is everything about what you're supposed to know for a healthy diet in terms of carbohydrates. So yeah, and this is the summary. Oh yeah, this is a concept uh, from one of our biology performance tasks. Sugars are converted into two things: reducing sugars and non-reducing sugars. So this is the Benedict's test principle. These are called amylites, the banana extract, which is a reducing sugar, and the alkali, which is basically the Benedict's reagent, containing Cu raised to 2 plus. I think this is cupric, and Cu plus is basically cuprous.